streaming from the American College of Cardiology virtual conference, the late-breaking trial in Mavacamten. I have the sessions going on here on my other screen, so excuse the very rudimentary manner in which I am broadcasting today. I am not a professional broadcaster. I'm just the CEO of the Hypertrophic Cardiomyopathy Association, so I'm doing my best. I'm trying to listen to two things at once while social distancing during COVID-19. So we're making do. Oh, that's funny. There's a doctor in the background as somebody else is talking here. I'm going to bring them into the live stream in just a second. Once they finish up, they're finishing up a previous session on anticoagulation. And we have a physician on right now who has somebody. This is kind of funny. I'm going to put this up just for a little bit of a joke here. Okay, I like this guy in the background here. He has no idea he's on camera. Whoever you are, dude, peace out. This is funny. That is very funny. He's answering questions about an anticoagulation study. So I see some of you are joining us now. Welcome. We're going to be live in just a few moments. I'll put the volume up on the session when it is live. My apologies for my timing error before. This session of ACC started at 10:30, but the HCM portion isn't until 11:30, and they're running a little bit behind schedule. Some technical issues and some just typical conference issues. They normally run a little bit behind. So there's a gentleman speaking right now from, I'm not exactly sure, an Asian country. That's all I'm sure of. I forget where he was from. But he had some really interesting data on valves, and it was which anticoagulation is better. And I believe that they came up with warfarin was still optimal in the first 3 months. So there you go. There's a little bit of data there. We'll be watching both screens. Hello, everybody who's joining us. If you have any questions, you can post them on the side in the comment section. I will not be able to address any questions regarding the study. It would be inappropriate for me to do so. If you do have any questions after the study is presented, please put them in the comment box, and I can circle back with the investigators and ask the questions and see if I can get answers. But I can't address those questions live with you here today. Possibly later in the week, I'll see if I can get one of the investigators to join us to follow up on this. Hadn't thought about that until just now, actually. So let's figure out how much longer this guy's going to talk about anticoagulation before we can go live with the data from the clinical trial on Mavacamten, which was conducted by Myocardial and a team of investigators from around the globe. And we're excited to look at that data. Many of you who are viewing actually participated in this clinical trial. Thank you so much. What's interesting is most patients don't get the opportunity to be part of the trial and then actually see the data in a late-breaking presentation that would appear normally behind closed doors at a major medical conference. But you're going to be able to peek in on that in just a moment. If they would stop talking about anticoagulation. But they'll get there in just a moment. Okay, let's see. It looks like he's wrapping up. Nope, a few more seconds. We'll get there. And dun, 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 dun. So if you want to look at the title of this talk, you will see all of the investigators that participated in the creation of the data that you're going to be seeing. Some of them are doctors' names that you're familiar with, and some of them may not be familiar to you, but they're familiar to me. And I know that the team worked very hard to get this data put together, and it would be, it's going to be really interesting to watch it live with you guys, so you can comment on the side. Again, I can't answer questions directly, but if they're not answered in the presentation and you want to ask a question, put it in the comment section and I can circle back with the investigators and see if I can get your questions answered. Oh boy, this guy's still going. And we're almost there, guys. Hang with me. It's coming, I swear. Okay, let's see.
And trying to be a little bit provocative, the assumed event rate in the control.
This is Marty Leon.
The warfarin arm was pretty high.
He's the moderator of the session.
And you selected a non-inferiority margin of 8%, which is a very, very broad margin, with an unequal.
Give him a little what for on his data.
Experimental arm, which allowed you to be able to have a small sample size in a randomized trial. But I must say that if we're going to change the guideline recommendations and begin using a DOAC, post-valve therapy, we need more data. We need larger numbers of patients to be assured of safety and to be at least somewhat confident that there really is an efficacy benefit relative to no therapy, which is generally the standard in the United States. So I wondered if you'd reconsider and because you are in an ideal setting to do a larger study in Korea, because this is the standard of care, because the rest of the world is looking for larger randomized.
That was kind of funny, wasn't it?
With DOACs and valvular heart disease.
Yes, I agree with you.
Okay, we're still talking about anticoagulation. Oh, Korea. He's from Korea.
A lot of limitations using the warfarin because of our diet is there's a lot of beans and a lot of problems in the warfarin. So there is some limitations using the warfarin in the early post-operative period. So we started this study, this kind of concept. So I think, of course, the 8% of the non-inferior margin is very broad range, but at least I think in the previous, we want to find.
We're going to have to edit this down, I think.
Reference for the non-inferior margin, but I think it's event rate in 12% in the warfarin group and about 4% in the edoxaban group in the previous study. So I think 8% or 7% is optimal for the this kind of sample size. But I think this is starting.
We'll be with the HCM data in just a few minutes. This is this talk in front of our talk. And they're wrapping up, and he's making his final arguments for his paper, which is looking at NOACs and regular warfarin anticoagulation. And he got questioned a bit, and that's what we're watching right now. So this is what happens in scientific sessions every time I've ever been to one. Yeah, get the experts questioning the younger guns, if you will. And this is Marty Leon, and he's a very well-known cardiologist, and he's kind of schooling the guy from Korea a little bit. That's the way I read it.
We're going to move on now to our last presentation.
And here we go.
Session. Sometimes we say the best for last. This is, again, a very provocative, interesting study that is entitled Safety and Efficacy of Mavacamten in Symptomatic Non-Obstructive Hypertrophic Cardiomyopathy, the MAVERICK-HCM study. This will be presented by Carolyn Poe from Brigham and Women's Hospital in Boston.
Here we go.
Well, good morning. And on behalf of the MAVERICK investigators, I thank the American College of Cardiology for the opportunity to present the results of the MAVERICK trial in non-obstructive hypertrophic cardiomyopathy. My disclosures notable for are notable for receiving research support and consulting honoraria from Myocardial, the sponsor of this trial. Classically, we think of hypertrophic cardiomyopathy as a disease with dynamic left ventricular outflow tract obstruction. However, at least a third of patients with HCM have non-obstructive physiology defined by a peak gradient of less than 30 millimeters of mercury at rest or with provocation. Non-obstructive HCM is especially challenging to treat, and there are no proven or approved pharmacological therapies. Symptoms are thought to result from diastolic abnormalities and potentially inefficient myocardial energetics. In contrast to obstructive HCM, where surgical myectomy or alcohol septal ablation can reliably improve symptoms, for patients with non-obstructive HCM who develop symptoms refractory to medical therapy, cardiac transplantation may be the only option. Thus, there is a great need for effective medical therapy for these patients. By altering the contractile mechanics of the cardiomyocyte, myosin inhibitors have the potential to modify pathophysiology and improve symptoms associated with non-obstructive HCM. Mavacamten is best known as a novel drug being developed to treat obstructive hypertrophic cardiomyopathy. It is a first-in-class selective allosteric inhibitor of cardiac myosin, which reduces the number of myosin-actin cross bridges and thus decreases the excessive contractility that is characteristic of hypertrophic cardiomyopathy. This results in improving obstructive physiology. However, there is also experimental evidence that MAVA may also have properties that help to improve myocardial relaxation and myocardial energetics, two other hallmarks of hypertrophic cardiomyopathy pathophysiology that may be particularly relevant to patients without obstruction. Therefore, it may be beneficial for those with non-obstructive HCM. MAVERICK-HCM was a randomized, double-blind, placebo-controlled, dose-ranging study of mavacamten in symptomatic non-obstructive HCM. The primary goal was to assess safety and tolerability, and there were a number of exploratory efficacy endpoints, including change in NT-proBNP levels, peak oxygen consumption by cardiopulmonary exercise testing, and assessing NYHA functional class. Participants were randomized 1:1:1 to mavacamten at PK-adjusted doses targeting either approximately 200 nanograms per mil in group 1 or 500 nanograms per mil in group 2, or placebo. They were treated for 16 weeks, followed by an 8-week washout period, and then an end-of-study visit at week 24. All participants were started on 5 milligrams of mavacamten. Dose was adjusted once at week 6 based on PK determined at week 4. And this dose was maintained throughout the end of the treatment period at week 16. There was no opportunity for down titration based on clinical response, including left ventricular ejection fraction. There were several pre-specified stopping criteria, including if LVEF decreased to less than or equal to 45%. 59 patients were randomized, 19 to the group 1 mavacamten of 200 nanograms per mil, 21 to group 2, therefore 40 receiving mavacamten total, and then 19 to placebo. Baseline characteristics were quite similar across the treatment groups, with a mean age of approximately 54 years. A bit over 50% of the study cohort was female, and most participants were NYHA class 2. Placebo randomized participants were slightly more likely to be NYHA class 3 and also had a slightly lower peak VO2 at baseline. Baseline echo parameters were representative of a non-obstructive HCM population with a mean maximal LV wall thickness of approximately 20 millimeters and an LVEF of approximately 67%. So now let's look at the safety data. Here's a table summarizing the serious adverse events experienced by study participants. Mavacamten was generally well tolerated by most participants. The overall rate of serious adverse events was low, but twice as high in the placebo arm versus those receiving active treatment. Most serious adverse events were cardiovascular in origin, including recurrent atrial fibrillation and atrial flutter. Although the rationale of administering mavacamten to patients with non-obstructive HCM is to try to improve left ventricular relaxation and myocardial energetics, given that the mechanism of action of mavacamten involves decreasing contractility, it was important to closely monitor the impact of the study drug on left ventricular ejection fraction. And here you can see that there was a mild reduction in mean left ventricular ejection fraction of approximately 4% in the pooled mavacamten group compared to a reduction of 2% in the placebo randomized group. However, in this dose-ranging, PK-driven study, there were 5 participants with a documented left ventricular ejection fraction less than or equal to 45% who met pre-specified stopping criteria. The nadir LVEF in this group ranged from 38% to 45%. 3 participants had asymptomatic reduction of LVEF, which recovered towards baseline by the end of study at week 24. 2 participants had symptoms. 1 required hospitalization, but in this participant, this also occurred in the context of recurrent rapid atrial fibrillation. His symptoms resolved after stopping mavacamten, and his EF recovered after managing atrial fibrillation with cardioversion and ablation. Now that we've looked at the safety data, let's turn our attention to the efficacy results, which were performed in an exploratory manner. Looking first at NT-proBNP levels throughout the study, we see a statistically significant decrease in NT-proBNP levels for those treated with mavacamten compared to placebo. Placebo-treated participants are shown in this dotted line and overall showed a 1% reduction in NT-proBNP levels throughout the duration of treatment. Mavacamten-treated participants are shown either in this light blue line for group 1 or the darker blue line for the group 2 patients. And you can see that their NT-proBNP levels decreased by approximately 50% by week 4 and stayed at a lower level throughout the study period. In the group 2 patients who underwent further dose titration at week 6, there was a further decrease in NT-proBNP levels at week 8, consistent with a dose-dependent response. After treatment is stopped at week 16 and drug is washed out, NT-proBNP levels then increase back to baseline by week 24. These results are notable because BNP has been shown to be an independent predictor of morbidity and mortality in hypertrophic cardiomyopathy. These promising results are the first demonstration of medical therapy improving NT-proBNP levels in patients with non-obstructive HCM and suggest physiologic benefit from mavacamten, potentially in improving cardiac wall stress. Next, we examine changes in cardiac troponin I during this study. This graph focuses on the 1/3 of participants who had a baseline troponin level over the 99th percentile. Participants receiving mavacamten are again shown in the solid line with those received in either group 1 or group 2 pulled together, and placebo is shown in the dotted line. There was a statistically significant decrease in troponin levels for participants receiving mavacamten, but not in those receiving placebo. These results are also notable because troponin levels have additionally been correlated with prognosis in myocardial fibrosis and hypertrophic cardiomyopathy. We also explored the potential impact of mavacamten on functional capacity as assessed by peak VO2 on cardiopulmonary exercise testing and on symptoms as assessed by NYHA functional class. This used a functional composite endpoint that is being used as the primary endpoint for the Explorer trial, the pivotal phase 3 trial for obstructive HCM that is currently ongoing. We are encouraged by these data because they suggest that non-obstructive HCM patients with more severe disease manifestations may benefit the most from mavacamten. Although there was no significant decrease in, or there was no significant achievement of this functional composite endpoint in either the intention to treat population or those who received per protocol treatment. Patients in this combined subgroup, which is defined as either having a troponin greater than the 90th percentile or E over E prime ratio greater than 14 at baseline, 1/3 of these patients showed an achievement of the functional composite endpoint compared to no patients on placebo, again suggesting that patients with more advanced or severe disease expression as monitored by troponin and E over E prime ratio may have more of a benefit from receiving mavacamten. So here are the key takeaways from the MAVERICK study. In this phase 2 dose-ranging study in patients with symptomatic non-obstructive HCM, mavacamten was generally tolerated by most participants. Reduction in LVEF can occur, and administration will be guided by clinical parameters, including LVEF and serum biomarkers. This was the first study to show an improvement in serum biomarkers of hemodynamic stress and myocardial injury in patients with non-obstructive HCM. There's also a suggestion that patients with more severe disease may benefit the most from mavacamten. These results are supporting further study to better characterize the use of mavacamten in non-obstructive HCM and potentially in heart failure with preserved ejection fraction in general. I also would like to acknowledge the clinical trial sites, the site investigators, and all the staff for their hard work and contributions to this trial. And most of all, we are profoundly grateful for the partnership of our patients and families who enabled this important study to occur. Thank you. Dr. Leon, you might be on mute.
Dramatic pause.
Am I still on mute?
You're good. Whoops, you went back on mute.
Dr. Leon, if you could, oh, you're good. Never mind.
Okay, sorry. Well, first, again, I was complimenting you on a beautiful study, and I wanted to ask Marty Marian if he could provide some perspectives on MAVA use in non-obstructive HCM based upon these phase 2 results.
Thanks, Marty. So first, congratulations. Obviously, it's great to see clinical trial work, new drug applied to the non-obstructive cohort of patients with this disease. As many know, most of the attention over the last several decades has been really focused on improvement of outcomes in obstructive HCM. I also want to start first with one point in that regard is that the treatment goals for the non-obstructive HCM patients are predominantly improving symptoms, daily symptoms, and functional capacity, not mortality or hospitalizations, because those event rates are very low in the non-obstructive group, which makes a non-obstructive HCM different than the traditional HFpEF group. With that said, a couple comments about the study for Carolyn. It's pretty clear that the drug mavacamten had an effect at lowering biomarkers. But what was a little frustrating, I think, to see was that didn't translate into a clinical improvement with either improvement in functional capacity or NYHA class. It's starting to look a little bit like what we have seen in HFpEF, what we talked about actually to start the whole session off with Paragon study, in the sense that decreasing biomarkers in that group may not ultimately translate into a clinical improvement that we would expect to see. These data although preliminary, start to sort of suggest that too. My first question for Carolyn then is, do you agree? Do we have the right clinical endpoints really for this type of population with VO2 and NYHA class? Should we be looking at different ways of assessing success of therapeutics in the non-obstructive group? And my second question involves the important issue of safety. As Carolyn pointed out, about 12% of patients here had really a striking reduction in ejection fraction, ranging from anywhere, an absolute reduction in EF from 17 to 35%, showing a lot of individual variability in response to the drug. And so that raises concerns, I would suspect, about the translatability of this drug were approved into the clinical practicing arena. And two, I also noticed that there were a couple of patients who had decreases in EF in which the ejection fraction did not come back to baseline after the 8-week washout period. Is that suggesting that there could be a signal here of some adverse remodeling related to the drug?
Okay, great. Thank you, Marty. I'll try to take those questions in turn. I think first you were asking about endpoints and whether, and how do we actually monitor for success in these types of trials and in this patient population. I think that's a key question that all been struggling with. First of all, this was a phase 2 exploratory dose ranging study, so it was certainly not powered to assess clinical efficacy. So we thought it was quite promising and encouraging that there seemed to be a biomarker response, suggesting that there was physiological improvement in the patients that were treated with mavacamten. Now, certainly, we don't want to just have a drug that only improves biomarker levels without other more impactful results in the patients. So I think that's going to require further study to try to sort out, how we monitor for success, and how to best help our patients with endpoints that will assess functional capacity and symptom burden, including patients' reported outcomes. In terms of safety, we do know that mavacamten, its mechanism of action is to decrease hypercontractility and to decrease contractility. So there will be an effect on left ventricular ejection fraction. I think one thing that's important to recognize that this was, again, a dose ranging study where dose was adjusted purely by looking at PK. There was no opportunity per protocol to down titrate the dose in response to subtle changes in left ventricular ejection fraction. So in practice, and certainly in future clinical study, we will have to proactively monitor clinical parameters, including left ventricular ejection fraction and biomarkers, to ensure safe and effective dosing.
Carolyn, I wanted to follow up on Marty's question. This was not a particularly symptomatic group. Most were functional class 2. The use of beta blockers was at about 2/3, calcium channel blockers, and really a minority of patients. So I'm not sure that it's so easy to show much of a clinical benefit when you start in patients that are not that symptomatic. As you go forward with a MAVA study in non-obstructive HCM, are you going to target a similar patient population, or are you going to be looking for more symptomatic patients?
Yeah, I think that's a great question. I think that's one thing that, the other intriguing exploratory finding from this study, I think, was seeing that patients with more severe disease expression seem to benefit the most. So I think that, those are important considerations to think about whether to target a more symptomatic population and to also use other metrics of disease severity as guidelines to target those that we anticipate would benefit the most from this type of treatment.
Does anybody have any other questions or observations? Well, then I want to thank you, and I want to thank Rick, of course, for moderating this session. This was a fascinating opportunity for us to be able to get in. Okay, well, there you have heard the data as it was presented at the American College of Cardiology conference. You heard it in real time with everybody else. This was a safety study, so we're not looking at, just to go over what that actually means, you're not looking at the functionality as to whether or not this is going to be an answer for patients in terms of therapeutics. What we're looking at this point in the clinical trial process is the safety of this particular agent for people with HCM, and did it appear to be safe? And the data does appear that it is a safe drug in that space. The data that was presented that showed some patients had atrial fibrillation. We know that a certain population is going to have atrial fibrillation anyway. So if anybody's thinking that this drug may have caused the AFib, I don't know that we have enough data to say that at this time. And in my gut, I would say no, probably not. It's probably more so related to the underlying physiology rather than the drug. If anybody has any questions about this presentation, you can email the office. I will put an email down here in the comments, and you can give us your questions at 4hcm.org. And we will process those questions with the study investigators, and we will get back to you with answers if we can. When you're in a study and you're doing trial work, some of the questions can't be answered yet, either because the study wasn't powered to answer that question, or it's embargoed information for a period of time while they're still working on those answers. So if you'd like to ask a question, you can email it to the office and we'll be happy to help you.
I am going to get off to another phone call right now. I will be back at 3 o'clock this afternoon with Harry Lever, who will be discussing generic drugs and things that all patients should know. So please join us back here at 3 o'clock and have a great day. Stay safe and wash your hands.